Hello. Today I want to talk about alcohol use disorder and look at the different types of genes involved to see how they play a role in the disease. Alcohol use disorder, AUD, is a global health concern affecting millions and contributing to serious diseases and early mortality. Advancing research into its biological mechanisms can lead to better diagnosis, treatment, and public health outcomes. Alcohol use disorder is the excessive use of alcohol, disregarding the side effects. Repeated use of alcohol can cause one to increase the usage to get the same effect or have symptoms of withdrawal. 28.9 million individuals aged 12 and older have suffered from AUD in 2023, according to the National Survey on Drug Use and Health. Some of the symptoms include craving alcohol, unsuccessful attempts to cut down on alcohol, developing a tolerance, etc. Oxidative stress can also occur, which results in an imbalance of radicals in one's body, leading to a heightened risk of cancer or cardiovascular diseases. Heavy drinking includes more than 5 drinks in a day for males or more than 4 drinks for females. Alcohol dependence may be due to hereditary reasons, where an individual has a family history of alcohol, since around 50 to 60% of AUD is transferred from parents to their children. Furthermore, having mental disorders also increases one's risk of developing AUD. Individuals with mental disorders are twice as likely to have AUD as individuals without mental disorders or stress. Psychotherapy is an option for treatment where an individual can be exposed to counseling that will help them reduce the usage of alcohol. Other options include support groups led by trained professionals in which an individual can express their emotions and experiences, and 12-step programs that add sponsors in addition to support groups to help them aid in reducing alcoholism. AUD is personal and a serious condition that affects individuals' day-to-day lives and impacts their decisions. For clinicians to measure an individual's recent drinking patterns, state markers are useful tools that indicate a history of heavy drinking and recent drinking volumes. An example of a state marker is the volume of red blood cells that are used to calculate alcohol intake. A large molecule made of carbohydrates and protein called gamma glycosaminoglycintransferase (GGT) acts as a state marker since it's involved in the digestion process and located in hepatocytes. An increased in level of GGT in blood gives signs of early liver disease, usually due to increased drinking in individuals, through a blood test. This needs improvement since elevated levels can be noticed in an individual who has diseases related to their digestive system, like prostate disease, and only 30 to 50% of heavy drinkers show an elevated GGT. Carbohydrate deficient transferrin (CDT) is an example of a biomarker that is responsible for carrying iron throughout the bloodstream. CDT levels in blood are increased in individuals considered heavy drinkers, since it disrupts the attachment of celiac acid. CDT is a reliable marker found through a blood test, but it contains challenges since an individual can sometimes have a high CDT regardless of their alcohol consumption. Both of the markers also have low sensitivity towards AUD diagnosis, which would hinder their accuracy. Recently, fatty acid ethyl esterase (FAEE) markers have been used to identify heavy drinkers from social drinkers. FAEE has the potential to be a sensitive biomarker after follow-up confirmations, but it doesn't necessarily clarify the severity of AUD. In addition to small molecule detection in blood, differentially expressed genes and biological processes have been used as biomarkers of AUD for diagnosis purposes. Gene expression research was used to find drug treatments to treat alcohol use disorder through the nucleus accumbens and the prefrontal cortex brain regions. A better understanding of the biological changes of AUD may provide better diagnosis tools and tailored treatment options in the future. So for the research objectives, I hypothesized that after analysis of the dataset, there would be differentially expressed genes, DEGs, between the control and AUD groups, which can provide insight into biological processes. Next, I will look at the key genes and biological pathways involved to see how they correlate to alcohol use disorder. AUD matters since it is a global neurological issue and affects millions. So understanding its biological mechanisms can improve diagnosis, support effective treatment, and reduce alcohol-related effects. So for the dataset overview, gene expression omnibus (GEO) was used to find a dataset with a GEO accession number of GSE 62699 that contained 36 samples, of which 18 were patients without alcohol dependence and the other were alcohol dependent. The platform was GPL571. Expression profiling by array and Homo sapiens conditions were applied before picking a dataset. The data came from postmortem brain tissue samples that were obtained from the Australian brain donors programs. The inclusion criteria for alcohol dependence cases in the original study were binary and based on a physician's diagnosis following the DSM-IV criteria. Individuals in the control group consumed alcohol but did not meet the DSM-IV criteria. The research only used mRNA data. Samples were collected from the gene expression omnibus and analyzed through gene expression omnibus 2R, GEO2R. GEO2R was then used to analyze the samples to see a difference in expression between the two groups, which are the control and the disease. A reliable dataset was identified by clear separation between the statistically significant, that is, those that had a p-value of less than 0.05, downregulated and upregulated genes. The table on the right shows the candidate genes and their corresponding false discovery rate, along with their log-fold 2 change. So a volcano plot was established, and it was used to show differential gene expression between control and disease groups. Since the upregulated red dots and downregulated blue dots have a clear difference, this dataset can be used to find genes that contribute to biological processes in alcohol dependence, providing evidence for my generated hypothesis. The black dots represent all genes with a p-value of greater than 0.05, and they're not considered since they're not statistically significant. Highly upregulated genes correlate with higher log2 fold change values, while highly downregulated to lower log2 fold change values. So outlier analysis showed that sample GSM 1531650, which was in the disease group, could be treated as an outlier since its range was lower than Q1 minus 1.5 times the interquartile range and was highly expressed on the clustered heat map. So from the clustered heat map created, sample GSM 1531650 stood out the most since the gene expression patterns for this patient were unique among the samples of the dataset. Here, the clustered heat map and the box plot can be used together to find outliers in the dataset. In the box plot shown, the outlier is highlighted in a black box. So other samples varied slightly in length, but the difference were minimal, so they would still be considered a part of the dataset. Most of the samples had gene expression levels that were within the -2 to +2 range, indicating that they were similar. However, the outlier sample contained more genes with z-score with an absolute value of greater than 2, suggesting a hard difference in expression. Uniform manifold approximation and projection, which is UMAP, was used to see whether expression patterns differed significantly between control and AUD in this dataset. You observed partial, though incomplete, segregation of control and AUD samples, indicating differences in gene expression between groups on average, with significant within-group variation. So for the clustered heat map, the pHeat map R package was used to create a heat map, highlighting the difference in expression for the control and disease groups, making it easier to decide whether expression patterns differed significantly between controls and the AUD in this dataset. Firstly, the series matrix TXT supplemental file was uploaded from GEO and read to insert it into the data. The sample IDs from the GEO2R process were identified and put into groups, respectively, along with combining them afterwards. Then, the top 250 most upregulated genes with a p-value of less than 0.05 were selected to be included in the pHeat map. This was done by reading the file from the top, so the most upregulated genes would be selected first. The pHeat map package was installed and ready to be used. pHeat map of the selected data was then run, along with not showing the row names since there would be too many in the figure. Finally, column IDs were clustered and samples were added to the row names to create a clustered heat map for the samples. The use of ggplot2 was done to create the graph by combining the independent components. So the clustered heat map showed all of the samples from the dataset and their expression. This map was created by using pHeat map package R to find any samples under or overexpressed depending on their color on the map. So higher expression is represented by red, while lower expression is represented by blue. So the sample we talked about earlier, the outlier, sample GSM 1531650, is highly expressed and can be treated as an outlier, as also seen from this figure. The outlier can be further seen in the dendrogram since it shows the control and AUD samples clustering together, apart from the sample I considered as an outlier, which we can see in the boxed sample. So string DB. This is the map of the most upregulated genes after analyzing with StringDB. It was created by plugging in the top 250 most upregulated genes with the p-value of less than 0.05 into StringDB by selecting multiple proteins, and the species chosen was, of course, Homo sapiens. The map was simplified by increasing the minimum required interaction from 0.4 to 0.7, excluding the disconnected nodes from the network. 8 candidate genes were selected from the StringDB since they're related to at least 2 KIT pathways. So for the age-rate signaling pathway and diabetic complications, we first initially hypothesized that after analysis of the dataset, there would be differential gene expression between control and disease groups, indicating that expression levels for significant genes vary, which can provide insight into biological processes. After further analysis of the groups through GO2R, it was deemed that the groups differed in gene expression, as indicated by the volcano plot and clustered heat map. Differential gene expression analysis revealed enrichment of several biological pathways, significantly the age-rate signaling pathway in diabetic complications. The pathway is known to be involved in the regulation of oxidative stress and inflammatory responses. The selection of this pathway as highly enriched amongst the differentially expressed candidate genes suggests its potential relationship in AUD, which is alcohol use disorder. In the age-rate signaling pathway, multiple candidate genes were selected due to their upregulation in the differential gene expression analysis, including RELA, which is NF-kappa B, it's a subunit of NF-kappa B, IL-1R1, STAT3, COL4A1, COL4A2, and TNF-RS-F1A. These can be possible regulators of oxidative stress and inflammatory responses. In particular, RELA can regulate gene expression by affecting promoter accessibility to transcription factors and also play a role in many biological processes like immunity, cell growth, apoptosis, differentiation, and more. In T cells, it is essential for cytokine gene expression and appears to be involved in the invasion-mediated activation of IL-8 expression. Research involving humans illustrated that alcohol dependence can lead to a loss of peripheral T cells, disrupt their functions, affect the activation of T cells, and disrupt the balance between different types of T cells. Other cases show similar increases in RELA, one being the function of immune cells and metabolism. In this situation, RELA restricts oxidative phosphorylation from turning to glycolysis. It promotes the expression of genes under oncogenetic stress that favors the M2 activation, which is an anti-inflammatory activation along the mitochondrial oxidative phosphorylation. In alcohol dependence, excess consumption can lead to inflammation, affecting the M2 macrophage and causing further issues for the individual. So, the upregulation of the candidate genes suggests a relationship between AUD and increased inflammatory responses along with oxidative stress. Oxidative stress from alcohol may lead to an activation of the inflammatory responses, while increased inflammation can lead to oxidative damage, representing a potential feedback relationship between these processes. Alcohol can also stimulate NF-kappa B activity and cytokine expression in the brain, which is seen through the dataset since it was derived from the brain biopsies. The increased levels in the samples could serve as NF-kappa B as a mediator of addiction-related gene expression. GGT and CDT were not seen in the differential gene expression, possibly due to them not being highly expressed in the postmortem brain tissue compared to the control. Both GGT and CDT are considered to be low sensitivity, with GGT being 61% and CDT ranging from around 26 to 86%. This generally has more variability. Additionally, the CDT marker results in a significant rate of false negative results, while the GGT marker can be affected by certain medications, along with pancreatic or prostate disease in an individual. Overall, the KIC pathway analysis suggests that AUD may relate to the symptoms of oxidative stress and inflammatory responses through the age-rate signaling pathway and the upregulation of the candidate genes, providing evidence for the original claim. This illustrates that an increase in alcohol may be associated with higher levels of oxidative stress and inflammatory responses due to the activation of age-rate signaling pathway in diabetic complications. So, some limitations include that the dataset contained 36 samples, so an experiment with a larger sample size would be more reliable and could also provide more evidence. Another key limitation is seen in the tissue, specifically in the data. The samples were derived from nucleus accumbens. However, this limits the detection of common blood-based biomarkers such as GGT and CDT. These markers did not show significant expression in the dataset, one reason being that they're not highly expressed in the brain tissue. Additionally, both biomarkers had low sensitivity and can be influenced by other certain conditions. RELA is involved in multiple biological processes, so isolating its specific role in the context of AUD would require further analysis and modification. This leads us to a critical question for future research. Can RELA be proposed as a biomarker for alcohol use disorder? So here are some of the references of my research, and thank you so much for listening.